Hi, this is Mr. Matthews. These are your notes on cell membrane. Please follow along with these notes. These are your Cornell notes. Make sure that as you write down the information, you're also answering the questions to the left. And at the very end, you write a 2 to 3 sentence summary of what the notes were about. So this is all about the cell membrane. I want to go over some of the terminology that we talked about in the Bubble Lab. And again, just, first off, just saying that, the cell membrane does separate itself from everything else, the surrounding environment. The whole idea is that it's going to control or even regulate. Remember, we talked about that term regulate, the traffic in and out of the cell. It's selectively permeable, allows some substances across easily, while others need some help. And that it's made out of phospholipids, proteins, and other things like carbohydrates. So that's sort of what we're going to talk about. I know in the Bubble Lab we mentioned those terms fluidity, we mentioned the terms flexibility, we mentioned the term selective sealing, and the word transport protein came out to really specify how the cell membrane actually functions. So taking a look at, we want to start with just the phospholipids themselves. So phospholipids are made up of a phosphate and a fatty acid, a lipid. Here's a picture of it, and I drew this on the board when we were talking about the bubble layer. And the phosphate group is hydrophilic and polar, meaning that hydrophilic meaning that it's attracted to water, and of course polar molecules attract to water themselves. And that's great because we need the cell membrane to be attracted to things. We want things coming in the cell, we want things to be leaving the cell. So we look at the lipid tails, and lipid tails are hydrophobic, meaning that they're nonpolar and they're going to repel water. So we can't have too much coming in, we can't have too much going out, otherwise the cell's going to get bigger and explode, or the cell's going to shrivel up and die. Either way, that's not good for you. So we have to have some way of regulating, again, what's coming in and out. And with this structure itself, you're able to really control that and make this membrane semi-permeable, meaning only certain things can come in and certain things can go out. I'm going to look at the arrangement. It's arranged in what's called a bilayer, a phospholipid bilayer that looks like this. We have molecules on the top and molecules at the bottom. So, again, one of those structure and function examples. So looking at that arrangement, if we want to really delve into it, the phosphate heads are on the outside because we want things to be attracted to the cell membrane, and on the inside they're facing the inside of the cell because we want things to be attracted to the inside so we can move things in and out with ease. And the lipid tails are the hydrophobic inside, and again, that if anything kind of slips through that doesn't need to get through, it'll be repelled to the outside. So it's really important that the bilayer is structured this way. That outer membrane of the cell, here's a picture of the cell under the microscope, and that, remember, every cell has this structure as part of its cell, the cell membrane, and it's made up of all these neat features. The lipid bilayer, you can see in this picture we have some proteins in it, and we'll talk about that just a little bit in general. But again, looking at that phospholipid bilayer, double layer membrane, you can see these molecules are going to be moving around. That's why it's fluid, and that's how it's flexible. Okay, remember, some molecules can pass directly through and just kind of spread these molecules out enough so they can get through. Things like gases, carbon dioxide and oxygen, can freely flow in and out through this membrane. And in order for that to happen, again, these molecules need to be moving and fluid and flexible to get these gases in and out. Again, just a reminder that the top is, I can't stress this enough, the hydrophilic heads are polar, and the nonpolar area are those hydrophobic tails. Looking at just two really important characteristics that I've stressed previously, again, because of this makeup, because of the phospholipid bilayer and the polarity and the non-polarity of the membrane, it creates situations where things are, or the membrane is selectively permeable, regulating what's coming into the cell and leaving the cell, and that helps maintain that word that we highlighted on the board, homeostasis, maintaining that balance. We can't have too much coming in or too much leaving, or that's going to be bad for the cell. The membrane is fluid. It's flexible and able to move and change shape. and that's due to those molecules. It makes it kind of bendy, but we obviously need it to be somewhat rigid, and that's where the proteins and the carbohydrates come in to help kind of stabilize it. So it is flexible, but it's not going to be flexible to the point where it's going to fall apart. So here's this is the staple picture of what it looks like, and you can see here we often call the cell membrane a fluid mosaic model, where it's just kind of a collage of all sorts of different things. Like you see these, you have the phospholipid bilayer, you have cholesterol that helps it stabilize, you have proteins, you have glycolipids, which glyco meaning there's some sugar involved, and all sorts of things. There are different types of proteins, which we'll get in a minute, and here we simply have what's called a transport protein. The other types of proteins, the three that we'll really highlight here, we have receptor proteins, we have marker proteins, and we have transport proteins. Transport proteins are really like a tunnel or a passage. They're going to allow things to get through. So going back to the bubble lab, right, your transport protein was that string. When you put that string on the bubble and you popped a hole in the middle, that was your transport protein. You could move things in and out of that bubble, and that was really good. The receptor protein is going to transfer information from one side or the other. So for instance, you read in the case study about diabetes and how diabetes is controlled by insulin, a hormone. Well, that insulin, the hormone, comes and binds to a receptor protein on the cell membrane, and that says, hey, cell, open up, let sugar in. And when that happens, you get the sugar to come in the cell, the sugar goes through, you go through cell respiration to make ATP, and you get energy out of that. If insulin is not available or not able to bind, then the sugar is going to have a hard time getting through that cell without the door being open, allowing it to come in. So that's going to be an issue. That's why people with diabetes, it's a really serious issue, and they have to take synthetic insulin. They give themselves basically a shot to give themselves insulin, and their body is able to open up that transport protein to let that sugar come in. Lastly, you have marker proteins. Marker proteins identify you as a typical type of cell. For instance, in our red blood cells, we have these marker proteins that identify yourself as an A blood or B blood. If you have O blood, you don't have the protein, but they just simply are what they say they are in the definition. Marker protein. They mark, they identify the cell, and that helps you communicate from one cell to the other. When we talk about how specifically things move in and out of the cell, that's called diffusion. And in typical diffusion, you can see in the picture here, we're moving from an area of high concentration to low concentration. So I'm going to back up a bit, and you can see this this diagram again, where you have all these little molecules here that are going to be crowded in one space. And generally, things don't like to be crowded, so they're going to move out naturally to where things are evenly spaced. That's what it means to move from high concentration to low concentration. So let's back up. You see high concentration, low concentration, and natural movement of molecules across space. It's like if I walked into the room and I sprayed some cologne, like lots of cologne or lots of Febreze. Someone standing in the back of the room might not smell it initially, but eventually those molecules would spread out evenly to where you could smell it. We talk about diffusion in that way as well when it comes to smelling things. Diffusion is what's called a type of passive transport, where it requires no energy to do this. Right? Molecules are going to naturally move from a crowded to less crowded space, and that's a good thing. If we needed to move from the opposite way, a less crowded to crowded space, where we're going to make things more crowded, we would need energy to do that, and that's called active transport. But passive transport, we're just simply moving high concentration to low concentration. It's called with the concentration gradient, until equilibrium is reached. Right, diffusion across the membrane. Again, we have that cell membrane that's separating the cell, and it can be penetrated. So things need to come in, things need to go out. Things like food and carbohydrates and sugars and proteins and amino acids, they all need to go into the cell. Lipids need to go into the cell. Salts and oxygen and water, all good things need to come in. And things that need to flow out are waste, ammonia, salts, carbon dioxide, water. These are all things that need to move out. And again, when we talk about how they move, They typically move from an area of high concentration to low concentration, either in the cell or out of the cell. And specifically when we're talking about water, that's a special type of diffusion called osmosis. So if we look at diffusing through this bilayer, right, fats and other lipids can get through the cell directly, right? Sugars, nope, too big. Amino acids, too big. Too big. Water, too big. Salts, too big. Ammonia, too big. All right. The molecules that cannot get through directly are polar molecules, things like water. Right? Things that can are ions, salts and ammonia can't get through. Things that really can get through are, backing up a bit, things like lipids and things like gases, carbon dioxide and oxygen. So if the cell can't, or if molecules can't get through that lipid bilayer, like carbon dioxide, like oxygen, then they need some help, right? That's what's, the transport proteins help them get through that. And what you have to understand is that each protein is specific. Each transport protein is specific to what it's going to move in and out of the cell. For instance, you have in this picture, you have 5 different transport proteins, and they're a different color because they represent what it is that they're going to transmit in and out of the cell. For instance, sugar has its own transport protein. Amino acids have their own transport protein. Water can only fit through its transport protein. It's not going to be able to fit through the sugar protein. Right? Other things like salt and ammonia, right? They all need things to come. They all need help getting in and out of the cell. And we often call that facilitated diffusion when we need help getting in and out of the cell. Whenever we talk about transport proteins, it's a special type of diffusion called facilitated. We're going to come back to that in just a minute. So again, speaking of facilitated diffusion, all right, each channel is specific to what it's going to transport in and out. And again, the word facilitate just means to help. So simple diffusion might be moving gases in and out of the cell. Facilitated diffusion is going to be moving sugar in and out of the cell. When you eat food, that food has to go across your cell membrane to get energy from it. You need some help getting across your cell membrane. That's facilitated diffusion. If you want to move down in your notes at the very bottom or on the back, it says active transport. Just know that active transport needs energy. We're going to make things more crowded. Cells may need to move against the concentration gradient or more crowded. So we're going to use energy to do that, often called a protein pump. And we go from low concentration to high concentration. So if we sort of summarize all the types of transport, we have passive and active. Those are the two sort of major categories. Passive, no energy needed. Active, you need energy. When we look at passive transport, we're either talking about simple diffusion of those nonpolar molecules like oxygen, like carbon dioxide, across the phospholipid bilayer, or facilitated transport where things like sugar and amino acids and other larger molecules might need some help getting across the cell membrane. So you have a transport protein helping them. Then active transport, again, is moving from low concentration to high concentration. We need energy to make things more crowded. So here's our sort of our really good picture. There we have simple diffusion simply across the cell membrane. We have facilitated diffusion going through the transport protein. Those two make up passive and active transport. The difference here, we are making something more crowded. So we need energy to do that in the form of ATP. So water is a special case. Water is called osmosis, the movement of water in and out of cell. And it's special because we have special types of osmosis. And just for so by definition, we want to say that osmosis is just the diffusion of water from high concentration of water to a lower concentration of water through the semi-permeable membrane. So the concentration of water, the direction of osmosis is determined by comparing total solute concentration. So something like salt or sugar. And we're going to do, you might have done the lab already, the cell membrane lab that you worked on with the dialysis tubing. And those are examples of hypotonic and hypertonic solutions, which again, we are referring to a comparison of solute concentrations, not typically the movement of water. So in a hypertonic solution, we're looking at more solute, less water. And just get this definition down. We're going to do lots of practice with what this means, and I'll explain more about what this means. But just in general, we have three types of osmosis: hypertonic, hypotonic, and isotonic. And by definition, again, hypertonic, more solute, less water. I'm not asking that you understand this now. Just make sure you get this down, and I'll explain it when we are in class for review. Isotonic means that there's an equal amount of solute, an equal amount of water, and hypotonic means that there's less solute and more water. So if you look at this picture here, this is a hypotonic solution because there's less solute and more water. In the hypertonic solution, more solute, less water. It gets a little flipped when we start talking about where the water is actually going to go. So when we have this setup right here, this solution is hypertonic to the outside of the solution. And again, if that doesn't make sense, that's okay. We are going to review what these means later. I just need you to get down the definitions and know that they exist. The consequence to all that I'll share is that in hypotonic solutions, water actually moves into the cell. And if too much water goes into the cell, then the cell could burst. And that's a bad thing. In plant cells, the plants get more turgid, and that's just a measure of rigidity. In a hypertonic solution, it generally means that water leaves the cell and it can shrivel up and die. That's not good. Okay, for instance, if you eat a lot of salty potato chips, you're going to be thirsty because all the water from your cells left your cells. You put your cells in a hypertonic solution and the water left, and you need to rehydrate yourself. So anytime then you drink water, you're putting your cells in a hypotonic solution, meaning water is going to flow into the cell. But it really all goes back to counting percentages and the amount of solute that is in your cells. And again, we'll do lots of practice on that. But these are the three situations that make osmosis really special. Right, here we have an example. I am actually going to flip through this. We will talk about this in our notes, but I'm going to flip through this really quickly and just end with active transport. And so we've talked about diffusion, or we've talked about passive transport, moving molecules in and out with no energy, and active transport, moving molecules to a crowded space using energy. This is about active transport, moving large molecules in and out. What happens when we have really large molecules or waste that have to go in and out of the cell? And sometimes it requires that we use energy to do this through the form of vesicles. The two instances are called endocytosis and exocytosis. If we need to bring things into the cell, that's a type of endocytosis. It's almost considered cellular eating. Like if you bring things into the cell through a vacuole, that's endocytosis. When you need to expel things, that's exocytosis, right? Getting rid of it. Again, the waste moves through the cell through a vacuole and eventually is expelled through the cell membrane. This concludes our notes on the cell membrane. Make sure that you write your conclusion at the end and that you answer the questions.